Welcome. My name is Allie Depew, and I am here with Dr. Mike Kavanaugh today. Mike Kavanaugh is from the University of Montana, and he is the director for the Center of Structural and Functional Neuroscience. So we got a neuroscience scientist with us today. That's pretty amazing. This is the We Are Montana in the Classroom program. And so I'm going to, without further ado, let Mike take over, and I'm going to pop out of the way here.
Okay, thanks a lot, Allie. Can you guys hear me okay? All right, give me a thumbs up. If you guys have questions, raise your hand and we can open up the audio mic so we can hear each other. But you guys are the only class on our connection right now. We might be joined, as Allie said, by a couple more classes in a minute. So what we're going to do, what I'd like to do today is talk about your brain. And what we're going to talk about is the way your brain works. And the way we're going to approach that question is to talk about the molecules and the neurons that are in your brain and how they work to allow our brains to do the amazing things that our brains do. So, the subject of our talk, right here, it's not a very impressive object.
Not today.
So it's
Sorry.
The brain, no problem. The brain is about 3-pound blob of jelly. And what it does is a lot more impressive than the way it looks. And if you think about some of the things that your brain can do, it's actually kind of mind-boggling, pardon the pun. One quote I've put up here is from a neuroscientist at the University of California named V.S. Ramachandran. And can you see that whole quote? Am I blocking
You're blocking the quote. That's okay. We can just scooch over.
I'm going to move
over. You want to read it to us as well?
So he asked this question. How can a 3-pound mass of jelly that you can hold in your palm imagine angels, contemplate the meaning of infinity, and even question its own place in the cosmos? So, we might not have time to get to all these questions today, but we'll at least get to a point, I hope, where we can start to think about how the brain can mediate these incredibly complex activities. So, I want to start with an example of a really primitive organism that's going to, I hope, help us shed some light and understanding on how our brains work. Now, if we think about the topic of consciousness, something we might not think about that often because we sort of take it for granted. You wake up in the morning and you really don't think about your brain. Or does anybody think about their brain first thing in the morning? I don't. It's just kind of, the world appears when you open your eyes, and everything that you experience is coming into you, and we just take it for granted. So what is consciousness? If we really kind of step back and think about it, what does that really mean? What do you guys think of when you think of consciousness?
Being awake?
Being awake. Yeah. Kind of the opposite of the way you are in your first period class, maybe?
No.
No, of course not. Well, okay, consciousness is a little bit of it's actually a very complicated topic. But one way to kind of begin to think about it that I think is useful is thinking about it in terms of perception. Because perception is one of the things that our brains specialize to do. We can perceive the world, we can experience it through our sensory systems, our hearing, sight, smell, touch, and those mechanisms of our nervous system allow us to experience the world. So if we go back and look at a very primitive organism that's actually an algae, and we're going to consider a molecule in that algae that I think is kind of interesting, and it sheds some light on the way our brains work. So there's an algae called Chlamydomonas, and this organism swims through the sea And it has a little spot in it that you can see, that little green spot. It's known as an eye spot. And in that eye spot, there are molecules on the surface of the membrane that are adapted to sense light. And so when light shines on those molecules, they open a little pore in the membrane that lets ions flow through the membrane into the cell. And that allows the animal to detect where light is, and it actually lets it navigate and swim towards light. Okay, so that's interesting, but what does it have to do with your brain? Well, it's interesting because that molecule, that light-activated ion channel, is called Channelrhodopsin. And the gene that encodes that molecule, that encodes that protein in the algae, is a molecular ancestor of a protein that's in our brains. Can anybody guess what that protein might be?
We're no longer alone.
You're no longer alone. That's right. Hello, Moore. Welcome.
Hey, more.
We have Dr. Kavanaugh with us today, and we're talking about brain proteins at the moment.
Okay, more schools meet Charlo schools. You guys are both on.
How you doing?
All right, so Moore came in a little bit late. We just started, and we've been talking about the brain and the molecules in the brain that are important, that give rise to all of the abilities that your brain has. And what we're going to be doing is talking about how they work a little bit. We're going to go through a couple of examples and give you some insight into how your brain works. And so we started talking about a molecule called Channelrhodopsin. You can see it up on your screen. And it's expressed, this molecule is found in an ancient organism, a type of an algae that can swim. It's kind of on the border between animals and plants. It's a distant ancestor of us. Animals and plants diverged about 1.5 billion years ago, roughly, as far as we can tell from molecular archaeology that allows us to kind of analyze gene sequences and compare sequences and see how DNA changed over time. But what's cool about this molecule is that it's an ancestor of an important molecule in our own nervous system. And it's designed in the algae to sense light, as we talked about. So I was asking if anybody could guess what this molecule might be the ancestor of in our own nervous system.
Let's get a guess from each school. Charlo, what do you think?
We are lost.
Think about how do we perceive light?
Your eyes. Yeah, your eyes. Retina. Actually, it's our
Retina. Somebody said retina. That's great.
Great. Moore, do you have a thought?
Yep, go ahead, Tim.
The cone in the eye?
Cone in the eye. That's a really good answer. That's a type of a neuron that's in the retina. It's called a cone. And it turns out, you guys are all on the right track, that gene in the algae is related to the gene in our own eyes that's expressed in neurons in our eyes, like cones, like is in this picture from the cover of Science magazine. And the structure for that molecule was solved about 15 years ago. And now we know that this is where our molecular mechanisms came from that let us see. They evolved over billions of years of evolution, starting with these algae. So this is just one example of a molecule that we can kind of trace back in distant organisms, and we know is an ancestor to molecules that are functioning now in our own brains and give us the abilities to sense the world as we perceive it. Okay, I'm going to talk about an example of one other molecule, and then we're going to go into some of the interesting things that this molecule does. The next molecule. This molecule is called the glutamate receptor, and it's been termed a key for learning and memory. And we're going to talk about learning and memory, and we're going to use that as an example of one of the most important things that our brains can do, or the most important activities that our brains can do. And this molecule, the structure that you're looking at, this cartoon, is actually based on a very highly defined, very precise X-ray crystallographically analyzed atomic structure that was determined by a guy from Montana named Eric Guo. He grew up in 9 Mile, just outside of Missoula. And he's in Portland, Oregon now. And he solved a lot of the structures of molecules present in the brain. And this is one of the most important ones that he's ever found. And I think he might even win a Nobel Prize one day for this work. But we're going to go into a little bit about how this molecule works in your brain and how it helps us to learn and remember. But first, we're going to take a little detour and talk about a patient who has been called the most famous patient in all of neuroscience. And when I was studying neuroscience, I went to college and I majored in biology, and then I got interested in neuroscience in college, and I decided to go to graduate school to study neuroscience in the brain. And when we were taking neuroscience courses, and everybody that studied neuroscience, there was a patient who went by the initials HM. And we all learned about HM because what he taught us about memory and learning was fundamental and transformed our understanding of how memory works. He died about 7 years ago, and now we know his name was Henry Molaison. So the initials HM were always used in books to protect his privacy. But his relatives had granted permission to reveal his identity, and his brain was donated to science, and his brain's been dissected, and it's still being studied. So what made him such an interesting character? It turns out that when he was a little boy, he had a bike accident. He fell off his bike, and this was back in the around the turn of the century, before anybody wore bike helmets. And it's a good lesson too that we should all wear bike helmets, because when he crashed and hurt his head, he ended up developing epilepsy, which often happens after a traumatic brain injury. And his epilepsy was incurable. And it went on for years and years, and his doctors that treated him were completely unsuccessful at treating his epilepsy, and it became uncontrollable, and his life was so miserable that it was decided to try a radical surgery to remove a part of his brain that the doctors had mapped and knew that was undergoing some aberrant activity, and they decided to take out this region of the brain. And the region of the brain that they took out is called the hippocampus. And on the left side of your slide, you can see a cartoon of a normal brain, and on the right side is a cartoon of HM's brain after the surgery. And during the surgery, there was a very precise removal of the exact borders of this brain region called the hippocampus. Now, what was remarkable was that after his surgery, first of all, he was cured of his epilepsy, which was great. He didn't have that problem anymore. He was able to walk and talk and get along fine. But something else happened that was remarkable, and that was that he was no longer able to form new memories. He could remember things that had happened in his life before the surgery, but he was completely unable to remember anything that happened to him as he went through life. So the neuropsychologists that studied him, when they realized that he had this very particular learning deficit, he became a very interesting patient to study. And one of the people that studied him was named Brenda Milner, and she's just won a neuroscience award that's the most prestigious award in neuroscience, called the Kavli Award. She just won it last year, and she's still alive. She's 95 years old. But over many years, over decades, they published work about HM, and the fact that he couldn't remember anything was kind of an incredible phenomenon. She would see him virtually every day and interact with him, but every morning when she went in to meet him, she had to introduce herself. It was like they had never met before. So I'm going to skip ahead and talk about the hippocampus a little bit more in terms of what we know it does. But there's a lot of work that shows now that our hippocampus is a central part of our brain that's important for establishing new memories and learning things. I'm going to show you a little movie of a mouse brain that features the hippocampus of the mouse, and it's using a technique called confocal fluorescence laser scanning microscopy. You don't have to remember that. But what it is a new technique that lets us visualize individual neurons by putting a fluorescent molecule inside these individual neurons. And so if we do that, we can kind of regenerate a three-dimensional picture, a map of the brain. And you can see here up on the top is the cortex of the brain, and this curved structure underneath it is called the hippocampus. And the neurons in the hippocampus are neurons that are involved in encoding what we learn and what we remember. And this kind of visualization technique is revolutionizing neuroscience because it lets us see neurons that we could never see before. When we looked at that glob of jelly that was a brain, it'd be really difficult to kind of get any kind of an understanding at a cellular level of what's going on without this kind of new technology. So what can we do with this kind of technology?
I'm wondering if we could maybe pause and see if anybody has any questions or comments on this, because that's some pretty incredible imagery that we just saw. What do you guys think? More? Do you have any thoughts or questions for Dr. Kavanaugh?
Is that a memory that we just saw?
I'm going to show you a memory in a minute. I'm going to show you what we think is a memory. But what your question is really great because it's sort of been the holy grail of neuroscience to understand what is a memory. How can we approach that question? And so what I'm going to talk about is how we do approach that question, how we can measure some functions of neurons that we think reflect what goes on when a memory is formed. Did you guys have any questions at Charlotte?
How would you get those scans, or how would you be able to, or where's that picture come from? Or like, how would you take that picture?
Yeah, so good question. I've kind of skipped over some of the technology because we don't have a lot of time, but it's new technology that lets us put a gene into a mouse brain that encodes a fluorescent protein, and that protein that glows when we shine a special wavelength of light on it, we can then detect that glowing using a combination of lasers and computers and imaging, and then we can reconstruct the three-dimensional image. It's kind of related to MRI. Have you guys heard of MRI, magnetic resonance imaging? It's a way, if you go into the hospital and they want to try to visualize something that's inside your body, the doctor might suspect you have a tumor or some kind of other problem. You can go into this machine and a radiation will be delivered, and the machine will be able to reconstruct the three-dimensional structure of your body. So without getting into too much detail of that, what it reflects is now we can look at single neurons with really high resolution, and we can understand some of the connections better and how they work.
I have a really brief question for you.
Sure.
So the images that we're seeing, how large is that area that we're seeing?
So the last thing we saw in that movie was of a part of a mouse brain that was about 3 by 3 by 3 millimeters, small cube of the structure of the brain.
So these are very tiny pictures.
Very tiny pictures. So this picture you're looking at here is another picture, actually from my lab, of a mouse brain using the same kind of technology. And you can see on the left side, the square is blown up on the right side. And that lets you see the structure of this neuron a little bit higher resolution. And if we go even to higher resolution and look down here at the dendrite, what we can see is kind of the structures that are in your handout. That you've got this handout of a neuron structure showing some of the basic properties of the neuron, the cell body and the axon and the presynaptic axon terminal. And what we're going to be talking about is kind of the coolest part of the neuron and how it signals. It's called the synapse. Does anybody know what the synapse is? Can anybody tell me what a synapse is?
Charlo, how about you? Do you know what a synapse is?
Yeah. Is that like
The space between the two.
Is it the space between two neurons?
Yeah, good. It's the space, it's sort of a gap between neurons, and it's key to how your brain works, and we'll see as we go along why that's true. But it's the place where neurons communicate with each other. It's like where they get really close together. They don't touch, but they're close enough together so that one neuron, that's called the presynaptic neuron, can talk to the postsynaptic neuron. And how do they talk to each other? How do they communicate? How do two neurons transmit information?
Moore, do you have a thought on that? You can guess too, it's okay.
Yeah, it's okay to guess. So, anybody at Charlo?
The chemical reactions or electronic sparks?
Yeah, okay, so there's an electric signal that comes first. It travels down the axon of the presynaptic neuron. And when that electrical signal reaches the end of the axon, it triggers the release of neurotransmitters. And the neurotransmitter molecules float across that synaptic gap, the gap between the two neurons, and the neurotransmitter binds to receptors on the postsynaptic neuron. And that's how the postsynaptic neuron knows that the presynaptic neuron is sending it a signal. And this gets back to remember the glutamate receptor molecule that we looked at. We're going to talk about how that receptor on postsynaptic neurons can receive signals that are sent by the presynaptic neuron when it releases glutamate. Glutamate's a neurotransmitter. Has anybody heard of neurotransmitters before?
Thumbs up if you have heard of them, thumbs down if you have not. What do you think?
Okay. Well, so some of you have heard of them, some of you haven't. Neurotransmitters are the chemical messengers, and there's over a dozen different ones that your brain uses. We're going to just focus on one of them called glutamate. Glutamate is the most important neurotransmitter in our brain because it is what communicates excitation. It's how neurons talk to each other and tell each other, okay, look, I'm sending you a signal and you've got to listen to this because something important is happening. Glutamate is released by the presynaptic neuron. It binds to receptors on the postsynaptic neuron, and then that allows the postsynaptic neuron to start to generate its own electrical signal that then leads to, guess what happens then?
What do you guys think? What happens?
If you think about where did the electrical signal come from in the first place?
I hear some murmuring. Charlo, what do you think?
I would guess.
There's a lot of
I guess, Allie, I feel compelled to point out that every thought they're having, whether it's boredom or excitement, is a result of this going on right now as we speak.
That's right. Virtually all your perceptions involve this process. So Well, the electrical signal that travels down the axon, causes release of neurotransmitter. Neurotransmitter then floats across and binds receptors on the postsynaptic neuron, and that triggers another electrical signal, and that can then get passed on again to talk to another neuron. And what's amazing in the brain is that this communication in synapses goes on in all the neurons in your brain. So here's a little bit of a picture that kind of talks about what happens when glutamate is released. At the terminal, you can see the axons and the little circles that contain glutamate as a transmitter. When the glutamate's released, it floats across and it binds to glutamate receptors. And one of the types of glutamate receptors is the one we're going to talk about that's involved in learning and memory. So synapses are really, really fundamentally important for the brain, for how it works. And the reason that's true is because no one really understands everything that's happening at the level of cells and molecules when we learn something. But there are some things we are beginning to understand, and one thing that's really clear is that synapses are plastic. And what do you think that means? If you were going to design a system that was a memory device, like a computer, what would you want it to be able to do?
Learn and change.
Learn and change. Somebody's saying, yeah. Charlo. Yeah, Charlo. More. What do you guys think? Come on, don't let him show you up. What happens in a computer when you store some information in the computer? What happens? What's going on? Every little register of memory in a hard drive or a computer memory device can be in one of two states. It can be 0 or 1. Usually that's how we think of it, as the state. And so we can encode information at points in the hard drive as 0s and 1s. And our synapses work in a very, very similar way. They can exist in two states. And we call the on state a potentiated state, and the regular state is just a normal state. So they're really a lot like, our brains work at least a little bit like computers. There's 100 billion neurons in our brain, which is a little bit hard to understand because it's a number that is really beyond our everyday experience. But what I'm going to tell you is something that's even kind of crazier, is that every neuron in your brain has about 1,000 synapses. That is, it forms connections with 1,000 other neurons, actually more than 1,000 other neurons. So, more. How many synapses are in your brain? If you have 100 billion neurons and you have 1,000 synapses per neuron, how many synapses are there?
A lot, I said. I see some people doing some quick calculations. It's a big number.
100 trillion?
Good. So yeah, 1,000 times a billion is a trillion. So there's 100, actually more than a trillion synapses. And if you think about, again, as a memory kind of register that can be in two states, 0s or 1s, the number of different states in our brain is equal to 2 raised to that trillion power. And that's a number that's like totally incomprehensible because it even exceeds the number of atoms in the known universe. But what's amazing about that is that it reflects something about our brains in terms of how much capacity we have and what we can potentially do. It's unimaginable.
So what you're saying right now is that the potential for our brains is nowhere near. We've not reached the potential for our brains. Is that sort of what that means?
I think that's a fair thing to say. I mean, you hear that sort of as an thrown off the cuff statement that we only use a fraction of our brains. But when we really start to dig in and look at the basic elements of the way our brains work, it's really amazing how huge the capacity is of our computing power.
My goodness. So what's happening right now in their brains? Just out of curiosity, like we're sitting here learning a subject that's probably new to most students, and so we're taking in brand new information. What's happening right now?
So we're taking in new information, and what I'm going to show you is how we think that new information is stored in the brain. And it's the way that works is by changes in the synaptic strength to go from this normal state to a potentiated state. So when you get excited, if I say something that's crazy or I start jumping up and down or yelling at you, you're getting more neurons activated, and some of them are going to get potentiated, and they're going to stay potentiated and remember things. I want to turn back for a second to something that is important to remember and think about. And at least for me personally, it's guided some of my motivation for studying the brain and learning and memory. And it has to do with what happens when things go wrong in our brain. And one of the most devastating diseases that afflicts humans is Alzheimer's disease. You guys have heard of Alzheimer's disease, and probably some of you, or many of you, might know someone with Alzheimer's disease. How many of you know someone, a friend or a relative that has Alzheimer's disease? A lot of you, and it's not surprising to me at all, because about 1 out of every 3 seniors who dies now in the United States dies with Alzheimer's disease or another neurodegenerative disorder related to Alzheimer's disease. And it's a debilitating disease that degrades our memories and really takes away our humanness, because it robs us of what we've remembered our whole life. And personally for me, my mother died of Alzheimer's disease, and seeing what she went through made me really think deeply about what we know and what we can do. And that's one of the areas of research going on now in neuroscience. One of the most active areas is Alzheimer's disease. And I want you to just watch this little movie. So, Alzheimer's disease is a disorder of our learning and memory, and we want to try to understand what's going on in this disease. We have to understand, first of all, how does learning and memory work normally? And then we can start to turn our attention to how we can treat the disease. So I'm going to tell you a little bit about how we can do that. How somebody asked before, can we see a memory form? And I'm going to show you an experiment done here by a student in my lab named Katie Sullivan, who's from Montana, and she was doing an experiment using a mouse brain to study learning and memory. And I'm going to show you the results of this experiment that she does to study long-term potentiation. And potentiation is what I was saying when a synapse undergoes this change from normal function to potentiated function. So can we watch a memory as it forms? So what Katie did was she's got this kind of complicated looking setup. It includes some electrodes and amplifiers that let her amplify signals, and she can record the electrical activity of individual neurons in the mouse brain that she's recording from. And this is a slice of the mouse brain. And if you look at the bottom, in the bottom corner over here, you can see that thing that looks like a little glass pipette. And that is a pipette, and it's touching that neuron. So she can record the electrical signals in that neuron. And what she did was, in another part of the brain, she gave a little electrical stimulus with a wire. And you can see that's what's going on here. There's this little artifact that's the electrical stimulation. And then that's followed by the electrical signal due to the glutamate receptors getting activated by glutamate. Now, okay, that is how neurons communicate. You can see that there's a presynaptic neuron that's being stimulated, and it's firing an action potential, and it's releasing glutamate, and the postsynaptic neuron is responding with its own electrical signal. Now, what's amazing is that we think, for us to learn, we have to be able to store information. And what happens is that following repetitive stimulation, if we do this at a high enough frequency and repeat it a few times, the signal gets bigger. And that signal, the glutamate signal, gets bigger for a long time. It stays bigger actually for as long as we can measure. And we know that it actually lasts for weeks and months and years. And in fact, it can last our whole lifetime. Because you might remember something that happened to you when you were little that you will never forget. And that memory stays with you all your life. And we think now that the molecular and cellular mechanism that we're storing these memories involves this kind of long-lasting change in the strength of the response of the postsynaptic neuron. Does anybody have a question about that?
I was watching a show the other day. I think it's called Unforgettable. It's a crime show. It's about a girl. It's based on a true story that she can't forget anything. How is that similar to this?
So that's really interesting, and it's a very rare phenomenon, but there are people that have completely photographic memories. And we don't really know what's going on, but there's some evidence that there are changes in the molecular mechanisms that underlie this process. Normally, it's not a perfect process. Our synapses can change and strengthen. Sometimes that strengthening goes away, it decays. Sometimes it persists for a long time. And there's a lot of room in the molecular mechanisms. It's a very complex process. We don't have time to talk about everything that happens, but we know that glutamate receptors in the postsynaptic neuron get moved from the cell body into the synapse. And they stay there, and because there are more receptors in the synapse, that neuron has a bigger response from then on, as long as those extra receptors are kept there.
So this is kind of like weightlifting with your muscles?
It's a lot like weightlifting. It's actually a lot like weightlifting. Using your brain, using your synapses, strengthens them, and the strengthened synapses persist for a very long time.
That was a great question, Moore. Do you have any other questions? Or Charlotte, do you have any questions at this point?
Or anybody have a question, go ahead and speak up. Yeah. Back in the corner and more.
Okay, yeah. So what is it that drives the memory then? The event or like something individual? What I'm thinking of is like sometimes two people experience the same event and someone remembers it and the other person doesn't. So.
Right. Yeah, great question. So if you think back again to the fact that we've got all these neurons and they're a very complicated series of networks and connections, when we perceive something, whether it's like we walk into our grandmother's kitchen when we're little and we see her face and we smell the soup she's making, and if it's something that makes an impression on us, that set of synapses that's stimulated by those perceptions, that stimulation is strong enough to make those synapses strengthen for years and years. If it's something that we don't really pay that much attention to, and maybe your friend walks into your grandma's kitchen, but really isn't paying that much attention to what's going on, though their neurons, their synapses aren't getting necessarily strengthened the same way. So there's kind of a combination of being exposed to a perception And also, it's not just the exposure, but it's the focusing on the perception and maybe the repetitive exposure to a perception. So when you're trying to learn something, you're trying to learn a math equation, you oftentimes need to look at that and read it over and over and over again. And but the more you repeat that exposure, the more ingrained it becomes in your brain, because your synapses become strengthened and solidified, and that experience becomes then ingrained like a memory. Does that answer your question? Any other questions? So aside from using an electrode to be able to look at to measure the electrical response and see how it changes, we can actually use that fluorescence technique and see how a neuron changes its shape after it's potentiated. And you can see this is a neuron, and up here is before learning and memory occurs, and then down here is after the event occurs. And you can see that these are the dendrites. This is a dendrite, and you can see there's a swelling that reflects all these receptors moving into that site. How's our time going?
We're going to be up about 15 minutes.
Okay.
And I'll leave some time for questions at the end, too.
Yep. Okay. So, what I'm telling you is, maybe it sounds plausible, maybe it doesn't to you. But neuroscientists really have been looking for this kind of molecular mechanism to explain learning and memory, a molecular and cellular mechanism. But to prove that really has anything to do with memory, we need to also be able to correlate it with real learning and memory, and show that it's something that does happen when we learn or remember. And we can use mice again. Mice are a great model organism because they have brains that are, of course, much simpler than ours, but they work fundamentally the same way. And there's a technique called a Morris water maze that we can use to study a mouse's ability to remember. And this is kind of like a spatial learning technique. It's kind of like an aqueous, a swimming pool with a maze in it. Because the mouse, the first time it goes into a pool, you can see that there's a little dark circle in the top right corner. The mouse can't see that, but that's a platform that it can get out of the water. It's trying to swim around and find a way out of the tank. And the first time it goes into the tank, it has no idea where that platform is. There are some cues, visual cues on each side of the walls that the mouse can see, but it can't see under the water. So we can put the mouse in this tank and let it swim around a while, and then the first time it gets exposed, it doesn't know where it's going. We can put it up on the platform, and we can just repeat this every day. And eventually the mouse learns where that platform is, so it'll swim directly to it. So this is day one, typical mouse, the way it behaves in this maze. Now, if we do the same thing every day for 7 days, and then put the mouse in again, the same mouse at the end of 7 days, watch what it does. It knows exactly where the platform is. So that mouse has remembered. It's been able to navigate its way by looking at the cues on the walls, and it can figure out where that platform is. And so we can use this kind of an approach to say, okay, well, what happens if the mouse has some kind of disorder or a mutation, or we give it a drug that affects its ability to learn? We can see how that affects its memory. So if we draw a little map of the way the mouse went on day one, you can see that it's kind of going around pretty randomly. Doesn't know where the platform is. It eventually finds the platform by chance. On day 7, it goes pretty much right to that platform. Now, okay, so we can. Do you think that this reflects some kind of form of learning and memory? Do you buy that? Has a mouse learned?
Thumbs up, thumbs down. What do you guys think?
Okay.
Yep. Unanimous.
All right, it's unanimous. Now, if we give the mouse a drug that blocks that glutamate receptor, doesn't allow it to signal, look what happens. The mouse is fine, it can eat and can behave normally, but it doesn't remember anything. After 7 days, it's pretty much at the same place it was on day 1. So this is an example of a kind of a behavioral experiment we can use to test ideas about the molecules involved in learning and memory. And so this and all kinds of other evidence supports this idea that the glutamate receptor activation is critical for learning and memory. Now we don't have that much time, but scientists have developed mice that have mutations in the same genes that we know are affected in some human Alzheimer's disease cases. In fact, there are a number of mutations in brain proteins that we now know lead to early onset Alzheimer's disease. And if mice have these same mutations, we can see impairments in this kind of a memory task in those mice. And we can also do the electrical recording and see impairments in that form of potentiation too. So why is that useful? Why would it be useful to have a mouse model of Alzheimer's disease?
What do you guys think? Charlo, yes.
So that you can relate it to your human life?
You can hopefully relate it to the human disease, and if the way the mouse behaves reflects what happens in the human disease, then what can we do with that knowledge? Yeah, somebody else next to you. Go ahead, Charlo.
A cure with it?
Cure.
A cure. There's the $64,000 question. Yeah, that's what is one of the most sought after cures in human medicine now, is Alzheimer's disease. And so using a mouse model of Alzheimer's disease to ask, what can we do, what kind of drugs might we be able to develop that will allow the mouse to not lose its memory, and then translate those discoveries to the human patients. And you're going to be hearing about this going on over virtually every week. You'll probably, if you start looking, if you start Googling Alzheimer's disease and research and Alzheimer's disease and cures, you're going to find that this is a very, very hot topic. And it's a very complicated topic. And I'm afraid that it's so complicated that it's going to take a long time to come up with a real solution to the problem. That's why I hope you guys get interested in the problem and you get interested in neuroscience, because we're going to need your generation to come along, and it's probably going to be your guys' generation that comes up with cures for Alzheimer's disease and lots of other neurodegenerative disorders like stroke and Parkinson's disease. The brain is the most complicated organ in our body. It's probably the most complicated structure in the universe. And it's not going to be something that we solve overnight. It's going to take a long time.
So is this something that's happening at the University of Montana right now, or?
So this is happening at the University of Montana and lots of other universities around the country and around the world. And in my lab, we're actually now testing some new molecules that have been developed at the pharmacy school at the University of Montana to see how they might amplify the glutamate receptor signaling and allow a better functioning brain and allow us to learn and remember more effectively.
Now I know we have just a few more minutes, so I do want to open it up to any questions for Dr. Kavanaugh before you have to change periods. I know some more students already had to go. Yes,
yeah.
More.
Would there be a way to artificially create a memory in someone else's mind or transfer certain memories into a different brain?
Yeah, that's a really interesting question. In theory, if we're right about how memories work, the answer is yes. And in fact, some experiments have been done with rodents that are kind of rudimentary experiments that are beginning to suggest that that actually can be done, because we can if a memory is pretty simple, we can understand the circuit of neurons that's activated during that memory. We can artificially activate that circuit, and we can in a mouse we can cause it to be activated, and we can cause it to be potentiated so that mouse remembers.
Mrs. Rasmussen to the library, please. Mrs. Rasmussen to the library. Thank you.
Charlo, yes.
Like on TV, you can see those guys who lose their memories for a couple of months or weeks, and then they just immediately come back. Is that real or not?
Yeah, that is real. There's, as you can imagine, the processes involved in memory loss are complicated too. Sometimes there are forms of short-term memory loss, and so networks that might reflect some memory gets disrupted, but that network isn't destroyed. It comes back after some time. That happens. There's longer-term amnesia, like with HM, where he becomes completely unable to remember anything new. But you might have experienced this if you've fallen off your bike, or you played football, or you got knocked in the head, that you might not. Oftentimes, there's a short-term amnesia that occurs after a concussion, and you might not remember what happened to you right before you got hurt. But then oftentimes that'll come back too.
Good questions. Yes. Joanne, yes.
Okay, there's a Alive Inside. Are you familiar with that work about music in Alzheimer's patients?
Yeah, there's a lot of interesting differences between the way we remember words and events and the way we remember music, because they're processed in different parts of our brains. And it's become clear that music can be a really effective therapy for Alzheimer's patients, because it can access different parts of the memory system that aren't as badly impacted, and it can kind of help them to remember different things that they couldn't normally remember. And we think that that's because, again, there really are very distinct circuits. Our brain gets compartmentalized in processing music versus speech versus visual information or things that we read.
Good questions.
Yeah.
Yes, Charlo.
So, do you mean music as the tones or the lyrics?
Both, really. The pairing of lyrics with the musical melody is memorized in a different way. It goes in a different pathway. It's stored in a different place. So you might, I don't know, how many of you have had an experience that you might hear a song, and when you hear the song, you can remember the lyrics. You can sing along with the lyrics. But if you just try to remember what the lyrics are without hearing the song or thinking about the music, it's much more difficult to remember the lyrics. So why that is...
Thumbs up, anybody have that experience? I know I have. Yeah.
So you said we could only use like a small percentage of our brain. So what would happen if we could use like all of our brain?
Great question. I mean, who knows really? I can't tell you the answer to that. You can see that there are people that have pretty amazing abilities to remember things, and oftentimes they need to. It doesn't just come naturally. You have to work and really strengthen your brain by repetitive exposure to an idea or to reading something. There's other things we know that help our memories and our learning and our brain's ability, that it's things that are really simple to do that have incredibly important effects on the way our brains work, and one of them is exercise. And we know now that just by getting out and exercising regularly, your synapses, we know this from looking at mouse brains, when mice exercise, they have way more synapses, and their synapses are more connected and healthy and function better. And the same thing appears to be true for humans too. So eating healthfully and exercising, which is really a boring kind of thing that you're told to do all the time anyway, does look like it really does. It does affect your brain's function, and it's a good idea to do that.
All right, everybody. Unfortunately, I have to end this. We have another group coming on at 2 o'clock, but I would like you to help me thank Dr. Kavanaugh today. And if
Thanks, you guys.
in the University of Montana as well. And if you have any additional questions about this, because this is a super fascinating subject, please feel free to email me. Your teachers have my email address, and I will certainly relay those questions to Dr. Kavanaugh. And again, of course, he's at the University of Montana, so come on down to Missoula and say hi. Thank you, everybody.
Thanks, you guys.
Thanks so much.